Okay, so I broke it down into cell cycle, genetics, protein synthesis, and then genomic technology. And then each part I've got stuff you need to know, like for identify questions and stuff like that, and then stuff you need to be able to do, right? So, for instance, for cell cycle here, this is stuff you should know. So you should know the different cell cycle checkpoints, right? You should know, for instance, that when the cell is in interphase, that it's going to start out by going through G1 phase, right? And then after G1, it's going to stop, right, before it goes into S phase, and it's not going to make more DNA yet until it's not going to make DNA until it checks to make sure that there are a bunch of things in G1 checkpoint. So it's going to say, hey, is there a need for more cells, right? Is this cell adhered to the extracellular matrix, right? Are there too many cells around? Is the cell density too high? And if those things are okay, then it will proceed and go through S phase. After it goes through S phase, it's going to go through G2 checkpoint, where it's going to say, hey, is the cell still adhered? Did we copy the DNA properly? That stuff is all in G2 checkpoint. And then those are all the checkpoints in interphase. And then it's going to go in either mitosis or meiosis. And mitosis and meiosis both have M checkpoint as well. And M checkpoint is going to be metaphase checkpoint, so it's going to check to make sure that the spindles are attached properly and that there's an equal amount of pull and push, and that's going to allow for it to pass M checkpoint. Okay, you should know what the DNA looks like at various times in the cell cycle. What's up?
Wait, are these slides going to be on the...
Yeah, I can put them on.
Okay.
Yeah. Okay, you should know what the DNA should look like in various stages of the cell cycle. So we know that in interphase, the DNA is going to be in either euchromatin or heterochromatin, right? And if it's in euchromatin, that means that it's nice and spread out, and that means that it's going to be able to be accessed. But if it's in heterochromatin, that means that it's going to be more compact, and so the promoters can't be accessed by RNA polymerase. And that's going to be only during interphase, right? Because that's when the chromatin is present. And then all the other stages, so prophase, metaphase, anaphase, telophase, those are all parts of mitosis and meiosis. And so during those parts, that's when you have chromosomes present, okay? And chromosomes are far too compact for any protein synthesis to occur. And you should also know what those chromosomes would look like. So we know that if we're talking about during prophase and metaphase, right? Prophase and metaphase of mitosis, or prophase metaphase of meiosis 1, that we're going to be looking at is the chromosomes that are the two sister chromatids, like that, right? Versus after metaphase, and so once we go through anaphase, we're going to yank them apart, right? And so they're going to be more like that, right? Both of these, all of these are considered chromosomes. These two together would be considered a chromosome, and then each of these is considered a chromosome. So if they're attached to each other, they're considered just one single chromosome, and then when they get detached from each other, they are separate chromosomes. Yeah?
When they're separate, can you still call them sister chromatids?
You still may call them sister chromatids because that tells you, we don't normally call them sister chromatids at that point. We just call them chromatids, right? Saying something is a chromatid denotes that it is just one full copy of the DNA. So it hasn't been synthesized, right? Okay. We know that, or you should know rather, that mutations can lead to cancer cells if they are in certain genes, right? Specifically in growth factor genes, which are also called proto-oncogenes. Right? Those are the genes that act like the gas pedal for the cell cycle, and they will cause it to, if they are overexpressed, they'll cause it to skip through all those checkpoints, right? And then the tumor suppressor genes, those are the ones that act like the brakes, and so those are the ones that trigger G1 checkpoint and G2 checkpoint. And so if you don't have the brakes, they'll just go right through the checkpoints, right? So if you have an underexpression in the tumor suppressor genes or an overexpression in the growth factors, you can, press the brakes a little bit, but then if you apply enough gas, you still move through it, okay? You should know that the process of mitosis is going to result in two identical daughter cells, right? And then meiosis is going to result in 4 unique haploid cells, right? So those 4 unique haploid cells are going to have non-identical DNA. They are those 2 processes that result in their non-identical DNA are the law of independent assortment and the law of crossing over. We'll talk about those, I think, next slide. You should know the differences between a haploid cell and a diploid cell, and you should also be aware of different types of cells above that, so triploid, tetraploid, and you should also know things like the even numbers of those, so it's a 2n, 4n, 6n, those can all be divided by 2, and therefore they can successfully complete meiosis, but other cells that are odd, so 3n, 5n, 7n, they cannot be evenly divided by 2, and therefore they cannot complete meiosis, but they can do mitosis no problem. Okay? Questions on things you should know here? Yeah?
Wait, so you can still have a 3N and then can complete mitosis?
3N can complete mitosis, no problem, because there's no formation of tetrads in mitosis. They're all just going to line up on the metaphase plate, so they don't have to have a homologous pair. But if it's 3N, then you'd have 2 homologous pairs and then 1 lone chromosome that can't form a tetrad.
Yeah.
What's up?
So, like, is that why, like, some cells would do, like, the meiosis instead of mitosis, or, like, the other way around?
So when we talked about, like, the bananas, for instance, we said that those bananas can't sexually reproduce, but they can asexually reproduce because they are able to go through mitosis, which is more of, like, asexual reproduction, but they can't go through meiosis, and therefore they can't sexually reproduce. They can't produce gametes, right? All right, so stuff you should be able to do in the cell cycle or along with the cell cycle is you should know what happens if the cell cycle is disrupted in any given stage. So errors during S phase, meaning that there's an error in the DNA. You should know that if there's an error during S phase, during the DNA replication, that it is going to try to repair that. And if it is irreparable damage, the cell will go through apoptosis. Right? You should know errors during meiosis for tetrads, right? So if you have the tetrads trying to form in a 3N cell, and so you can only form 1 tetrad and then you have 1 lone chromosome out there, that's an error, and that error will cause it to fail metaphase checkpoint, and so that cell would go through apoptosis. You should know failures during anaphase will cause non-disjunction, right? So something wrong with the spindle in anaphase would lead to non-disjunction. Non-disjunction would then cause there to be either a monosomy or a trisomy in the fertilized offspring, right? And then errors during spindle formation. So if the spindle can't form at all, some chemotherapy drugs disrupt spindle formation, and so they stop metaphase checkpoint completely. And then also, we talked about the changes in the growth factors and the tumor suppressors and how they relate to G1, G2, and G1 and G2 checkpoints. And then M checkpoint would be things like spindle formation, formation of tetrads that would fail that. Genetic disorders that are caused by aneuploidy when looking at a karyotype, right? So really the only ones that you're going to be responsible for knowing is if you looked at a human karyotype, if there were 3 21st chromosomes, that's Down syndrome. If there are there's 1 23rd chromosome, that's Turner syndrome, right? If there are 2 X chromosomes and a Y, right? That's Klinefelter syndrome, right? And so the X chromosomes you can tell because they're much bigger, so it would be labeled like 23, and then there would be like that, right? The Y chromosome is a little stubby guy like this, and so this would be 2 X's and a Y. That's Klinefelter syndrome. And then if you had 1 X and 2 Ys, that's Jacob syndrome. What's up?
So we have to know all the different ones.
Just those 4.
Okay.
So Down syndrome, Turner's, Klinefelter's, Jacob's. What's up?
Do we need to know what symptoms they
No.
have?
No, you just need to be able to identify, oh yeah, that's a trisomy of the 23rd chromosome, and there are 2 X's present, and so that would be Klinefelter's.
Okay.
Or in the case of 21, they would just all be the same. So 3, you have a trisomy in the 23rd or 21st chromosome, therefore that's Down syndrome, right? And you should also know that those are the result of non-disjunction. Right? Anybody have questions on the cell cycle? Anything you need to be able to do with the cell cycle? Okay. Let's talk about genetics. Stuff you should know. You should know all the processes that lead to genetic variation in sexual reproduction. So that's law of independent assortment, law of crossing over. Those, of course, refer to meiosis. Those are processes that happen in meiosis. Crossing over happens in prophase 1. Independent assortment happens sort of all of meiosis 1. Basic terminology for allele inheritance. You got homozygous means you have 2 of the same allele. Heterozygous means you have 1 of each allele. And then hemizygous means that you only have 1 copy of it total. So in the case of like sex linkage, males that have XY, if it's carried on the X chromosome, they would only have a single copy, a single allele, and therefore we call them hemizygous for that allele. You should know the probabilities for monohybrid cross. That's going to be 3 to 1 for the dominant to recessive phenotypes. And in a dihybrid cross, it's going to be the 9-3-3-1, where 9 means that it's going to be that they would show dominant for both traits. 3 would show dominant for one and recessive for the other. 3 would show dominant for the other one and recessive for the one. And then one of them would be recessive for both. Okay. You should know the levels of dominance. That's complete dominance, which means that there's no difference between the heterozygote and the homozygous dominant. They would show identical phenotypes. You should know incomplete dominance is when the heterozygote has a less defined version of the dominant phenotype, or a less extreme version of the dominant phenotype. So again, this was like we talked about flowers, where you could have a red flower and a white flower, and then when you cross them, you would have a pink flower, because the pink flower doesn't produce as much red protein, right, and therefore shows up sort of a lighter red color, which we see as pink, right? You should know all the extended genetic concepts. That's multiple alleles. So if you have more than 2 options, if it could be red, white, or there's also a yellow allele, that's multiple alleles. Polygenic inheritance means that there's multiple different genes that affect the single phenotype, right? So in the case of the height of humans, right, we said that it's not just one gene, and there's not just, 2 alleles for it, so you're not either short or tall. There's a bunch of different genes that have multiple alleles, and so you can have a whole range of height, right? Pleiotropy, that's when you have the opposite of polygenic inheritance. Pleiotropy is when you have a single gene that has multiple phenotypic outcomes. So albinism is a single gene mutation that results in changes to eye color, skin color, and hair color, right? And then epistasis is when you have one gene that overrides another gene. We talked about mice or rats, I think we were talking about specifically. We said that those rats could be black or they could be brown, but they could also be white, right? So they produce black or brown pigment, but there's a gene that controls whether or not that pigment is able to attach to the hair, and therefore, if they produce brown pigment that's not able to attach to the hair, it doesn't matter that they're ultimately producing the brown pigment, because they're going to show up as white mice, right? Sex linkage, we know that those are genes that are located on the X chromosome, not on the Y chromosome, on the X chromosome, and we know that females are then going to inherit 2 copies of them, and males are only going to have 1 copy, right? Yeah.
What's hemizygous? The one under the basic terminology.
Hemizygous means that they only have 1 copy. So in the case of sex linkage, if you are a male, and it's that allele is on the X chromosome, you would only have 1 allele. So if we did like big A, little a, that would be heterozygous, right? But in a male, so if that was in a female on the X chromosome, it would look like this, so we'd call it heterozygous. But if it was in a male, it would just be like this. So there's not a small a allele at all, right? Because there's nothing on the Y chromosome, and so they just have the 1 copy, and so they're not homozygous or heterozygous, they're hemizygous.
Is that also like complete dominance too then, since it's only like 1?
Not necessarily, no. It's not dominance at all, because it's not dominant over another allele. It's just the only one that's there. So it's the only option. Yeah.
What is the ratio? Like, what is each of the numbers?
So in a monohybrid cross, 3 means that there are 3 of the offspring that have a dominant phenotype, or 75% of the offspring would have the dominant phenotype, and 25% of the offspring would have the recessive phenotype, right? In the dihybrid cross, it's 9/16 of the offspring would have dominant for both traits. 3/16 are going to be dominant for one and recessive for the other, right? And then 1/16 are going to be recessive for both.
Oh, so both these are the same.
Both 3s are the same. It's dominant for one, recessive for the other, and then dominant for the other, recessive for the one.
And how would you do like a Punnett square? Like if you
You would not, never, ever. We'll get to that in the next slide.
okay.
Yeah.
For the Y, like, will it ever have like a little letter after it, or is it just always gonna be
No,
X?
it's always going to be nothing.
So like males are always gonna be homozygous.
Males are hemizygous if the trait is sex-linked, if it's on the X chromosome,
Oh.
right? Obviously, if it's on an autosome, a non-sex chromosome, then males have 2 copies of it,
Yeah.
right?
Okay.
All right. Yeah. Codominance is when both traits are shown equally, right? So, for instance, if we talked about blood types. So, if you are big A-O, right, you would have blood type A, right? But if you were big A, you would still have blood type A. So we know that A is dominant, right? But if you are big AB, then you are blood type AB because you're producing both protein A and protein B. And so one of them is not dominant over the other one. Both are dominant. Both are being expressed equally, right?
Wait, are you posting a video you talked about all this?
Yes. All right. So you should be able to, for genetics, you should be able to look at a cross between 2 individuals and the outcomes or the offspring that are produced by it in the F1 and F2 generation, and you should be able to hypothesize what types of interactions are going on, right? So the first thing that you're going to do when you do that is you're going to look for things that you know about, okay? If you're looking at a monohybrid cross, you should expect the offspring to be 75% dominant, 25% recessive, right? If you're looking at a dihybrid cross, you should look for the 9-3-3-1 pattern. Those are expected results, okay? Then, if you look and the results don't fit your expected outcomes, then you go through this list and you say, okay, well, if the males and the females show differing amounts of each of the traits, if there's an inconsistency between males and females, then what you're probably looking at is some sort of sex linkage where it's located on the X chromosome, okay? If you have offspring that are the same phenotypic combinations as the parents, right? Which means that, like, you know, let's say one parent was blue eye and blonde hair, and one was brown eye and brown hair, and then you have offspring that are either blue eye, blonde hair, or brown eye, brown hair, but they are not brown eye, blue eyes, or blonde hair, brown eyes, right? What that means is that they are linked together. And what linked means is that they're located on the same chromosome, and anytime you do see a new combination of them, so you do see offspring that are brown hair, blue eyed, for instance, that what happened is that there was a new gene linkage that was created by crossing over during meiosis, right? So again, you spot linked genes by looking at, or specifically autosomal gene linkage, by looking to see if the offspring have the same combinations of phenotypes as the parents, right? If you have a varied heterozygote phenotype showing up, so if you have, you know, a white flower and a red flower and it produces a pink offspring, right? That's weird too, right? That's different than what we should know. And so that's going to show you incomplete dominance. And then if you have phenotypes that appear out of nowhere, seemingly, right? So you cross a brown rat with a black rat and you get a white rat. Well, that's weird. That shouldn't really happen, right? So where does that come in? That's probably some other gene that's overriding that gene, and that's epiphysis, right? So these are just some quick ways that you can look at unexpected outcomes and determine what's going on in those unexpected outcomes. You should be able to analyze data using Punnett squares. You should be able to look at these these data tables here and draw a Punnett square that would explain why those things are happening, right? So that means that if it is sex-linked, you should use an X and a Y for the father and an X and an X for the mother. If it's autosomal linkage, you need to make sure that both of those traits are together in your Punnett square. So instead of doing one Punnett square for A and one Punnett square for B, trait A and trait B, you would do AB in each of the spots in just a 2x2 Punnett square, right? So in other words, if I had this going on, right, I would be tempted to do, okay, this is trait A and this is trait B, or this is trait A here, right? And then I do another Punnett square for trait B or something. That was bad. Right? So I'd be tempted to do this, but what I shouldn't do that. What I should do is I should combine these together and put this with this and this with this, right? When I do it, and then I run these through, and now I have big A, big B, little b, yada, yada, yada. And this would show that those genes are linked, and it's going to show the outcomes that we would expect from linked genes because they're being inherited together. There's no way for those gametes to separate, right? It says that you should only ever use 2x2 Punnett squares. So let's say that the genes weren't linked. Let's say you had trait A and trait B, right? And I said that the one parent was heterozygous for trait A, and the other parent was homozygous for trait A, and then let's do heterozygous for both parents for trait B, okay? And then you get asked, what are the odds that, or what percentage of the offspring would we expect to be to have the dominant A trait and the recessive B trait, right? Dominant A trait, recessive B trait. So we'd run these through. Okay, so what percentage has dominant A? 50%. And what percent has recessive B? 25. So how do you determine what percentage of the offspring then? You multiply those two numbers together. So what is 50% of 25? It's 12.5%. Right? So the combination of those two would give you the percentage that they would have both this trait and this trait if we multiplied them together, right? It's the law of probability. Right? And so this saves you from having to do a 4x4 Punnett square, because if you do try to do a 4x4 Punnett square where you foil the gametes, first outside, inside last, it's going to take so much time for you to do it, and you're going to run out of time on the test, whereas you can do a 2x2 Punnett square in almost no time. Questions on that? Yeah.
Would you have to draw like a Punnett square for like incomplete or epispace?
Well, you could. Right? If it was incomplete dominance, it would just mean that everything that was a heterozygote would have that third phenotype, right? The intermediate phenotype. And then if you were doing it for epistasis, you would have two separate Punnett squares. One for, say, if we were doing the rat example, one for pigment, and then one for pigment attachment. What's up?
Do you know if it's a 9, 3, 3, 1?
How do you know if it's 9331? What do you mean?
Like
if you, let's say I had two traits, A and B. Okay, and I said that in my offspring, I had, 98 that were dominant for A and dominant for B, right? I had 35 that were dominant for A and recessive for B. I had 31 that were recessive for A and dominant, or recessive for B and dominant for A. And then I had 12 that were recessive for A and recessive for B, right? And so now I chose really easy numbers here, but you could easily double them, right? You can make it 180 and 65 and 61 and 20 or whatever, and that would still be a 9331 pattern. But you can see here, you've got around 9/16 here that would be dominant for both. You've got around 3/16 that are dominant for one and recessive for the other, around 3/16 that are recessive for the first one and dominant for the other one, and then around 1/16 that are recessive for both. Okay, and these can be determined. Like I said, normally you would have to do a four box Punnett square, a 2x2, a 4x4 Punnett square in order to get these outcomes. But you can also use two 2x2s, because if you know that a monohybrid cross has the 3-to-1, if you do two 3-to-1s, what do you get? What are the odds that they have dominant for both? 3 times 3, which is 9. What are the odds that they're dominant for this one, recessive for this one? 3 times 1 is 3, right? 3 times 1 is 3. 1 times 1 is 1, right? So you get 9, 3, 3, 1 for those patterns by multiplying them together. All right, any other questions on this one, on genetics, things you should be able to do? Protein synthesis. This is probably going to be the bulk of the stuff on the test. You should for sure know how to do this really, really well. Okay, we should know the process of replication. Replication is making DNA from DNA, right? You should know the basics of the enzymes involved, right? You should know helicase unzips the double helix. Polymerase is going to add new nucleotides on, make a polymer, right? And then DNA ligase is going to join the fragments together. You don't need to know much more than that, though. You should know about transcription. Transcription is going from DNA to RNA, any kind of RNA, right? So you could be making rRNA, you could be making tRNA, you could be making mRNA, you could be making microRNA, right? But we know that specifically if it's going to go on to be a protein, it's going to be mRNA that you're producing, right? The rest of those kinds of RNAs, once you produce the RNA, that's the end product. So if you produced tRNA by the process of transcription, well, then you're done, right? It's going to remain tRNA. There's nothing else that's going to happen in that process, all right? You should know that after the mRNA is made, it needs to be modified, right? I guess I missed the first part. So RNA is going to, RNA polymerase, which is the enzyme that produces RNA, is going to bind to promoters. Those promoters are regions of the DNA that signify that that's the beginning of the gene, right? So they're going to bind to the promoters, and we talked about how you have euchromatin and heterochromatin. The heterochromatin is so tightly bound up, so jumbled up, that there's no open promoters, that the RNA polymerase doesn't have access to the promoters because they're all wound up. And so that's why that stuff doesn't ever get transcribed. And if it doesn't get transcribed, then it doesn't get made into a protein, and therefore it's not expressed, right? So those promoters are important to know, what they are, that those signify the beginning of the gene, and that's where the RNA polymerase attaches. You should know about mRNA modifications, right? So after the mRNA is made, it has a GCAP and a poly A tail added, and then it also gets its introns spliced out and its exons get spliced together, right? And once all those things are there, GCAP, poly A tail, and it's all spliced up, then we call it a final RNA transcript, right? And so that's what gets exported into the cytoplasm in a eukaryote. If it doesn't have those modifications, it's going to stay inside of the nucleus. All right. Translation is the next step. So once it goes out into the cytoplasm, it's going to get translated. How does it get translated? Well, tRNA is going to be the translator. tRNA is going to float around in the cytoplasm, and a specific tRNA molecule that has a specific anticodon on it is going to hook up with an amino acid, right? A specific amino acid. So there are a whole bunch of different tRNA molecules, because there has to be a bunch of them, because there's going to be one for each unique anticodon, right? Those anticodons all code for either a amino acid or a stop codon or a start codon in the case of methionine, AUG, right? And so you should be able to look at a codon chart. I guess that's on the next slide is that you should be able to use. But I guess on this one we can say that you should just know that tRNA has an anticodon that corresponds to an amino acid. Okay. You should know the different ribosome sites. So the A site of a ribosome is going to be where the amino acid enters. The P site is going to be where the growing polypeptide is held. So the amino acids enter through the A site, and then that amino acid gets added on to the polypeptide that's in the P site. And then the E site is after that amino acid is removed from the tRNA, the tRNA exits through the E site. And it doesn't have its amino acid on there because its amino acid has been deposited into the P site onto the growing polypeptide. Okay? You should know about differential gene expression. That's sort of the idea that every cell in your body has the exact same DNA sequence, but that they're all very different, right? All of the cell types are very different. How are they very different? Well, that's because in eukaryotes, you've got things like DNA methylation. Methylation is going to be where DNA becomes hydrophobic, and because it's hydrophobic, it's going to clump together to try to get away from the water, right? And then histone acetylation, which is going to cause it to sort of unwind more. And so methylating DNA is going to cause it to become heterochromatin. Acetylating histones is going to cause it to become euchromatin, right? Transcription factors, we know that that's how a eukaryotic cell controls what specific proteins it makes at any given time, are the transcription factors that are present. And then other things like post-transcriptional controls are going to be useful for temporarily stopping the production of a protein that's normally produced. And we have things like microRNA that would go and bind to the mRNA that would block it from being translated, and then repressor proteins that would bind to the GCAP and prevent the ribosome from attaching. Also on here is prokaryotic gene expression. Prokaryotic gene expression is controlled through the presence of operons. Operons are those segments of the gene where you have the promoter, and then you've got an operator, right? And that operator is going to block the promoter whenever it has a regulatory protein attached to it. Yeah?
What's the function of the GCAP?
The GCAP is where the ribosome attaches and where the small ribosomal subunit attaches to start the process of translation. And the GCAP is also necessary for the mRNA to be able to exit the nucleus, right? It's got to be able to go through those nuclear pores that are controlling what things go in and out. And if it doesn't have a GCAP, it can't get through the pores.
So it's kind of like the E site for the translation thing? Or things like things like exit after expansion.
Sure, yes, except for the fact that they're exiting through pores here, and they have to have that. It's like a pass, right? It's like if you have a backstage pass or something to a concert, well, they're not going to let you back there unless you got the pass. So the GCAP is a little pass that they show when they're exiting the nucleus through the nuclear pores.
Okay.
Yeah.
The operon is basically the start codon.
No. The operon is a bunch of DNA, right? A long sequence of DNA that has a couple different parts. It's got a promoter, right? Like every other gene does. But the key is that the promoter gets blocked by this other area that's called the operator, right? And that operator is going to have a protein that binds to it, and that protein is going to block the RNA polymerase from being able to move through and copy that gene. What's up?
Oh, you're good.
Did I get it?
Yeah.
Yeah, Chad.
What does the A, P, and E site mean?
A, amino acid. So amino acids enter. P, polypeptide is held. That's where the growing polypeptide is held. E site is where the tRNA exits after it's deposited its amino acid in the P site. Okay? So, stuff you should be able to do with protein synthesis, right? You should be able to transcribe a gene from DNA to RNA. This means you should know the complementary base pairs. You should know that when you're going from DNA to RNA, A goes to U, T goes to A, G goes to C, and C goes to G, right? Once you've transcribed it into mRNA, you should be able to then use a codon chart to determine the sequence of amino acids that the polypeptide will be, okay? So you're just going to find the triplets in the codon chart and then figure out which amino acid would be produced there. We know that it's always going to start with the start codon, which is AUG. You should be able to hypothesize how a mutation would change a protein's shape, right? So if you have a mutation that causes a polar amino acid to be replaced with another polar amino acid, well, that's not going to change the shape very much because it's not going to need to fold any differently because it's just one polar amino acid replacing another polar amino acid. But if you have a polar amino acid that is being replaced by a non-polar amino acid, right, that's going to cause a major change in the way that the protein folds because now the polar part, the part that was previously polar that wanted to be on the outside of the protein is going to now want to be on the inside of the protein because it's non-polar, right? What's up?
Amino acid polarity will be given to us, right?
Absolutely, yes. You don't have to memorize the polarity of amino acids. You should be able to relate protein synthesis to these terms, this term alleles that we used back in genetics. We should know that alleles are phenotypic traits. They're phenotypic outcomes that result from changes in proteins, right? And changes in proteins are the result of changes to the nucleotide sequence, right? So in the case of, we talked about the case of brown hair versus black hair here. So black hair is sort of like the perfect version of this gene that absorbs the most light. And so if you went through and you had, let's say, a polar to polar change in amino acids, right? That's not going to affect it very much, but it might affect it a little bit. And so that pigment might be slightly less effective at absorbing light, and therefore you might have brown pigment that's the result of a polar to polar change. But if you have a polar to non-polar change, that's going to change the entire way that the protein folds, and so that might be like a black hair to blonde hair change, or a black hair to red hair change. That's a much more significant change because there's a much more significant difference in the shape of the protein. Yeah.
So how exactly do you know if it's a polar to polar?
So on the codon chart, there are color coded to like they'll have different shades and say like these are the polar amino acids and these are the non-polar amino acids. And so if you know what the original one was and then you know what the new one is, then you can see like, oh, it's a polar to polar change or a non-polar to polar change or polar to non-polar or whatever, right? Yeah, there's also acidic and basic amino acids on there, but for our purposes, we know that acids and bases are polar, and so they're just going to get grouped into the same polar group. What's up?
But like the protein shape, well, like we have to say like it's bigger or smaller, just like
No, because it's really, really hard to predict things like that. You would just know like, oh, okay, that part used to be polar and now it's non-polar, so it's going to want to fold differently because the polar part or the non-polar part is going to want to be on the inside of the fold rather than the outside of the fold. Questions on stuff you should be able to do from protein synthesis. I guess down here in protein synthesis, we could also talk about incomplete dominance, right? So in incomplete dominance, when we talk about protein synthesis, we know that you're producing half as much of that pigment. So in the case of the red flower and the white flower making a pink flower, it's because now you only have one copy of the gene for the red pigment, which means that you're producing half as much mRNA for it, and if you're producing half as much mRNA for it, that means that you're producing half as many proteins for it, and half as many proteins for it will not get you as dark red of a color as you would if you had twice as many of those red pigments, right? And so that's what incomplete dominance, that's how incomplete dominance manifests itself with protein synthesis, right? All right, genomic technology, stuff you should know. You should know why we use the PCR. That's to create a bunch of different copies of the same DNA sequence. That's the polymerase chain reaction, by the way. That's what PCR means. You should know why we use restriction enzymes to analyze DNA, because if you're looking at just a big hunk of DNA, you don't really know much about it. So we use restriction enzymes to cut them up into different segments so that we can analyze them, right? Once we cut them up into different segments, we have to have a way of separating them into the groupings by how many nucleotides are in the segment. And so we use gel electrophoresis in order to do that. Gel electrophoresis utilizes electrical current. And so those electrical currents, they have a negative charge by the wells and a positive charge at the other end. And so we know that DNA is negatively charged, and so it's going to be pulled towards the positive end. Right, so it gets pulled through the gel towards the positive end, right? And we know that if you apply that same force, that electromotive force that's going through it, if you apply the same force to a whole bunch of different segments, right, then the segments that have the least mass will go the furthest, right? So, you know, obviously, you know, if I pushed my my cup here, right, it's going to go further than if I pushed my car with the same force, right? Because my car is very massive and the cup is not very massive. So I'll be able to move this a lot further than I can move my car. And so the mass of a segment of DNA is going to be based on the number of nucleotides in it. You should know what a transgenic organism is. You know the transgenic organism has had genes inserted into it from another organism. And you should know why we would do that, right? So in the case of farming, we would want to insert helpful genes that are going to code for things like antibiotic or pesticide resistance, and also genes that will make them grow faster, be hardier, taste better, things like that. Okay, and then you should know about the purpose behind the Human Genome Project. Really, all you need to know about it is that we wanted to know the sequence of A's, C's, G's, and T's in the entire human genome, right? And by doing that, if we do it to enough people, we can figure out what genes do what things by looking at, okay, all of these people have these genes that are matching, and then here's this person who has a genetic disorder, and they have this one change that nobody else does. And so at that point, we can start to target and say, okay, if you have this, then you have a higher rate of genetic disorder than somebody who doesn't have that gene. Stuff you should be able to do with genetic technology or genomic technology, you should be able to look at data from gel electrophoresis and tell what it means, right? So look at the bands that are created in a gel. I was just playing with them. Look at one of these, right? And you should be able to say, okay, well, these lines all match, and if these lines all match, that means that this DNA and this DNA were identical before they were cut up with the gel electrophoresis, right? But if you were to have different people, right, and I put the DNA from different people in these wells, then none of them would match. They would all have different bands because they would all be cut up into different segments based on the nucleotides that you have compared to the nucleotides that everybody else has in it. You should know about creating transgenic organisms that would be able to produce human proteins. Right, so we talked about E. coli specifically. We said that in order for E. coli to be able to produce insulin, a couple things have to happen. We have to isolate the mRNA from a cell that was already producing insulin, and then we need to take that mRNA and make it into cDNA, right? cDNA is complementary DNA, and then we have to insert the DNA into a bacteria or another organism that will then produce that protein. And we usually use viruses in order to do that. There are other ways of doing it, other what we call vectors for inserting cDNA, but you don't need to know anything about that. If we're going to use, the only two you need to know are if we're going to use a bacteria, we're going to use a plasmid in order to insert them, and if we're going to use another organism like a goat or something like that, then we would use a virus in order to insert a gene. And that's it. That's all. I mean, I won't say that's it because it's kind of a lot of stuff. But I don't think it's too overwhelming as long as you sort of break it down and like, okay, these are just a couple things that I need to be able to do. Does anybody have questions on genomic technology? Anything on this? Anything from the slideshow? What do you got?
Or you have to know the in detail.
Nope. Wouldn't worry about that. What's up?
So why? So bacteria use a plasmid, and then like a goat or like another organism who use like a virus.
Right.
But
That's
like,
because bacteria will accept plasmids. That's part of their normal thing that they do anyways, is they trade genes in plasmids. So if we put a plasmid around them, if you put a plasmid out, you know, in the space around a bacteria, they'll automatically just take it in and start expressing it. That's just something that bacteria do. It's called bacterial transformation. But a goat doesn't do that. If you put some random DNA inside of a goat's bloodstream, it's just going to destroy the DNA, right?
Oh, so you put the cDNA into like a virus and put it into the goat. Okay.
So then the virus infects the goat cells with this new DNA, right? So all those like gene therapy things that you read about in the book, that's how they do them, is by using a virus to infect the cells.
And how would you, like, what is exactly like a plasmid? Just like,
A plasmid is a small extra chromosomal ring of DNA, which basically means that if we know that a bacteria only has a single chromosome that's in a ring, right? It's just a really small chromosome that only maybe contains a couple of genes. Anything else? Yeah.